Mercury is a known neurotoxin. The main source of pollution is industrial waste, and as more and more countries became industrialized, the mercury pollution problem began to surface globally.
There are a lot of industrial processes that use mercury. As a catalyst, mercury is used in switches and thermostats. There are uses for mercury. And so any industry where mercury is being utilized, you have the potential for contamination from that industry. But a lot of the mercury that is emitted into the upper atmosphere is coming from the burning of coal. And because mercury is a trace contaminant in coal, and if there are not scrubbers or if there are not filters on the smokestacks to filter out the mercury, then the mercury is just emitted into the upper atmosphere, and the elemental mercury, the mercury that's at the zero oxidation state, will be transported globally.
Mercury contamination is not a new discovery in the scientific community. In fact, the most famous documented incident is probably the Minamata incident. It was discovered in post-World War II Japan.
The effects of acute mercury poisoning are devastating neurologically. You will get ataxia, meaning a hard time coordinating movements. It can affect vision, hearing. Basically anything that involves neurons is going to be affected, which is pretty much everything. It can cause psychosis, it can cause, and there's also documented effects of babies in utero whose mothers were exposed to these high levels of methylmercury in Minamata Bay. They also describe the cats, the feral cats that lived around the fishing wharves there would exhibit symptoms of acute mercury poisoning. It's a consequence of unregulated dumping of industrial waste, and the victims of that poisoning, some of them are still alive today. The biggest events happened in the 1950s, but there are still victims, a lot of this poisoning alive today, that are still, suffering from the symptoms of that.
In the country of Colombia, methylmercury is an environmental issue caused by the illegal and informal mining industry in the 21st century.
There is a vast use of mercury, and mercury is polluting air and water sources, and it's becoming a major increasing threat for the population. The problem is that the prices of gold are still very high. We're talking about $1,300 per ounce, and this becomes a very attractive business for all these miners, big industrial miners, small informal miners, and also the illegal gold mining.
How exactly is mercury used in the mining process?
The way it is used is that you build this amalgam between the ore that you extract from the mines, either through subsoil extraction or from the riverbeds, and they mix this with the mercury, and the mercury attaches itself to the gold, and then it is much easier to extract the gold from the ores. So with this, then you have this amalgam that has the clusters of gold with the clusters of the mercury, and then they burn this to evaporate the mercury, so that you end up with a pure solid gold. But then the mercury is evaporated into the air and is also released into the water sources, creating all the health threats that we know about. Because most of the use of mercury in Colombia for gold mining, it's informal and illegal, so it is hard for the governments and the organizations to follow up on this. But the suspicion is that it's pretty serious, that there is a vast amount of use of mercury in the extraction of gold around the country. And most of this extraction is happening in many areas where the government has difficulties in reaching out and doing the enforcement of the law. There are efforts here and there trying to eradicate the use of gold, of mercury into gold mining, and shift to use cyanide, which is less dangerous and it can be managed in a better way. So the regions in the state departments, they are trying to enforce some of these new technologies and trying to work with NGOs and the private sector and the miners to shift towards technologies that don't use mercury, or that use mercury with a technology that they can capture back the mercury without releasing it into the water or the air. These are what are called here retortas or retorts, which is a device that it's like an upside down bowl. So when you do the burning and the evaporation, you capture back the mercury again.
That's very
Methylmercury is a global problem and presents as an environmental issue here in the state of Minnesota as well. It has been shown that Minnesota is affected by methylmercury more than neighboring states.
So in Minnesota, there was a study recently published that they suspect that babies born on the North Shore of Minnesota are 1 in 10 of them has methylmercury levels that are of concern. And at certain methylmercury levels of cord blood, so when a baby is born, they can get an idea of how much methylmercury that baby has been exposed to by analyzing the methylmercury in cord blood. And 1 in 10 has methylmercury levels in their cord blood that are of concern, and there is a level of methylmercury. There's a level of methylmercury at which there are documented IQ effects that IQ can be diminished because methylmercury is a neurotoxin.
While methylmercury has been shown to cause neurological impairment, what makes the effect worse is its ability to bioaccumulate in tissue.
So bioaccumulation and biomagnification are processes that impact the distribution of a contaminant in an ecosystem. And so something that bioaccumulates, compounds that are lipophilic, meaning that they're fat soluble, tend to like to be dissolved in lipid-rich places. And one of those lipid-rich places is any kind of cellular structure. The cellular membrane is very lipid-rich. And so mercury will accumulate in the lipid bilayer of cells. So if you have a relatively low level of methylmercury just in a water system, you will get much higher levels in the organisms that live in that water system. The other issue with methylmercury is it is metabolized by organisms very slowly. It doesn't get metabolized well. And as you go up the food chain, these effects become more severe. So the organisms at the top of the food chain, the top predators, they're eating all the things below them that have eaten the food sources that have had methylmercury. And so as you go up the food chain, the concentration of methylmercury in tissues will go up by orders of magnitude.
The 2014 University of Minnesota iGEM team addresses the methylmercury contamination problem by designing a biofiltration system to filter contaminated water. The device contains many small silica-based beads, which is an application of the bacteria encapsulation technology.
In the lab setting, we're looking for things to be tested as quickly as possible. And one of the ways that we've tested these encapsulation systems is to just make simple gel plugs where we've got the silica material at the bottom of a glass vial, and it's in this silica that we encapsulate the bacteria. So there's silica matrix and a crosslinker that crosslinks the silica around the cell. We've got usually about a mil of this material down in the bottom of a vial, and then we add the buffer and whatever pollutant we might be testing on top of that. Usually 3 mils or so, up to 10. It fits nicely in the vials that we have, and then we incubate these together for up to an hour or however long we think it might take to remove the pollutant and measure the supername that's left. That any of the pollutant that hasn't been removed, we can measure that and get an idea of the rates, the reactions, and things like that. So it's a very nice system to test in the lab for lab scale. Okay, so as I was saying, then we often test these small gel plugs for lab scale, but what we envision is making silica beads that are about 1 millimeter or so in size to increase the surface area so that more of the bead can be in contact with the pollutant. These beads could then be packed into some sort of filtration system where you'd have water constantly flowing over the beads over time, the pollutant coming in contact with the bead surface and then the cells inside the bead. And the enzymes of the cells then would have a chance to act on the pollutant.
With success supported by our preliminary data, the team also examines the problem from the scope of GMO bioethics. We started by reviewing the ethical problems of GMO in a societal context.
GMOs, or genetically modified organisms, and sometimes people call them GEOs now, is genetically engineered organisms. And the reason for that distinction is because some people will say, well, you know, there has been a lot of genetic modification of all sorts of organisms that we've domesticated, of crops and of animals that are domesticated, that has been just through artificial selection, not through an actual engineering approach. And so genetically engineered organisms, most people are not concerned about genetically modified organisms that have been modified through the process of artificial selection. Genetically engineered organisms seem to be a lot more divisive in terms of public opinion. And that is, a genetically engineered organism is an organism where a fragment of DNA, a piece of DNA from another organism, has been inserted into the organism of interest. People have a lot of concerns about genetically engineered organisms. One of the concerns, well, there's a lot of concerns, and some of it is concern of the unknown, that this is a new technology, and that there is concern that there are going to be effects that we aren't predicting and that we don't understand. There are concerns because of the possibility of cross-pollination of genetically modified plants with wild-type plants. There are concerns because of potential health effects in consumers. There are concerns because people have ethical concerns about, should we be manipulating the DNA of another organism? So some people have ethical concerns in that way.
One major problem with assessing how the GMO affects the environment and local ecosystem is the duration that it takes to see an effect. The decision-making process should combine both benefits and harms, as well as providing flexibility to incorporate concurrent findings.
In general, whatever the benefit is should be weighed against the cost. That would be typically the most obvious thing to do, and the cost has to be an inclusive cost. So I don't think atrazine is actually the best example. The best example is a more surprising one, would be use of antibiotics. Use of antibiotics for people, that's got the biggest benefit. Use of antibiotics for agriculture, you know, we shouldn't use antibiotics for agriculture unless we have sick animals. It's a high cost of generating antibiotic resistance. Right. And so if we're not doing that for antibiotics, the likelihood that we're going to apply a kind of appropriate evaluation of costs and benefits for other things is more unlikely, because the benefits of antibiotics are so obvious. And so that suggests to me that we really have to have a more inclusive policy that really does weigh costs and benefits appropriately, and clearly we're far away from that now.
Thing. So that in the development you pay attention to those sorts of calculations, but you actually can't see everything during development. You can only see certain factors, and then policy is going to bring in a certain different set of dimensions for what needs to be evaluated, and then there might be pilot implementations where you get further information and feedback, and then you might evaluate both the development of a particular product or how the policy is structured. That, of course, means that it's not as simple as saying, yes, we decided it's safe, and now we can forget about it and move on to something else. I think that's probably what people would like. It would be nice, but when we're dealing with these, complex causal relationships in the world, that's not what we're going to generally find. So to follow up on Mike's example just a little bit more, the questions about antibiotic resistance that emerge from the use of antibiotics in different contexts and overprescription to some degree in medical contexts and their use in the agricultural setting. It's not just that you could have seen all of that, obviously, right at the beginning. part of it is reacting in real time to the fact, and now that we observe these things happening, it changes the decision-making structure, and we have to react to it and incorporate that into the decision-making.
A common GMO application is GMO food. GMO food has been shown to increase agricultural production by a dramatic fold, but the public opinion on consuming GMO food seems to diverge.
Well, I think the answer is that the public exhibits variation in how it responds to genetically modified organisms. For some people, it is relatively minor. It's not something that they spend a lot of time worrying about. For other people, any sort of tinkering with the food that is eaten in this sort of direct way makes them uncomfortable. And so between those relative extremes, you can find opinions all along the spectrum in between. I think that the concerns about the food would arise around several things. One basic feature would be safety. You know, is it safe to eat? And I think that in many cases, even in situations where we're not quite sure what the ecological consequences of genetically modified organisms, we have some good traction on whether it's safe to consume. But at the same time, I think people treat their food not just from the perspective of is it safe to eat, but also is the food that was produced, that comes to the table where I'm going to eat it, arising from sustainable processes and practices that are not simply oriented, say, towards making more money or producing large quantities of a particular food item. And so people bring different values to what they decide to eat, and I think that affects whether they, how they view genetically modified organisms. So that you could have somebody who is quite comfortable with the safety of genetically modified organisms, but still will not eat them for the reason that they think that the manufacturing and other processes that are involved in that generation of the genetically modified organisms is somehow problematic.
Controversies have not only arisen on investigating the effect of GMO, but also on other kinds of environmental pollutants. The methods that we use to provide persuasive and accurate data is therefore important.
A lot of concerns should be addressed with data. So I think showing safety, and there would be a variety of ways to show safety. You could show safety by showing that the organism that you've engineered is not actually going to be able to escape where a confined area. And so that would be one way of illustrating safety. You could demonstrate safety by basically doing sort of mesocosm experiments, where you look at the effects if the organism that you create, that you modify, is released into the soil. What effects may you see? 6 months later, is it still there, or has it been outcompeted by other bacteria? So I think a lot of these concerns need to be addressed by looking at data.
Scientific practice level, it's probably the case that for complex problems like the effects of atrazine, you really want to make sure that you are utilizing multiple methods to get at that you're not over relying on any one experiment or one type of measurement, regardless of whether it says there's a problem or not a problem, because that's likely then to not be as robust of a result in the long run.
Our evaluation of bioethics led us to reassess the ethical issue with our device implementation. We sought to alleviate this problem through initiatives to engage the public with basic science. In addition to empowering the public with knowledge, we also examined ways that we can address ethical concerns within scientific research.
One way is just to go make a field of genetically modified organisms and look at their surrounding neighborhood. The difficulty with that is that usually the consequences are small initially. And so small effects are hard to detect. And so if it's something that's cumulative, so this year it's a 1% effect, and next year it's 2%, and the following year it's 3%, you might not be able to measure any effect. And so as time goes on, it might be a consequence, but you might not initially detect that, even if you're very thorough about your mode of investigation. And so being conservative with respect to these things is often important, and mapping out a priori ahead of time where you might expect to see a consequence is critical for actually finding them and not just looking randomly, because looking randomly you're not going to see anything. You have to actually target your investigations carefully, and that means including people who are not focused on genetic modification, who are interested in ecological consequences, and oftentimes those are completely different researchers, have completely different research ideas and questions. Those are the ones that need to be in place, in addition to the folks who actually made or created the genetic modifications.
I don't know. I tend to think the appropriate question to ask is just standard risk analysis. I mean, do we understand the organisms we're putting into the lake well enough to be confident that we're not going to exacerbate the problem, or to just cause a whole another problem that's going to be as bad or worse, right? And if the products have been vetted, right, and if we know that they are safe or reasonably confident they're safe, to say, like, yeah, clean up the water. If you've got a better tool to clean up the water than existing tools, it seems to me neither here nor there that the new tool is genetically modified. The question is it safe, and if it is, then it seems good to use.
One of the ethical concerns is the scale of safely disposed bacteria-encapsulated beads and the genetically modified bacteria escaping our device. One precaution that we took in designing the device is to keep the bacteria in the silica beads and contained in the filter to prevent unwanted interactions within the local ecosystem.
In the laboratory system, we can just put that in the autoclave. And the beads, too, even on our lab scale, would be able to put that into an autoclave.
To address the scale in a device outside of the laboratory system, we also envision our encapsulated bacteria to have a kill switch system, which is a major future design to improve the biosafety of our device. Our next step is to address the bioethics and safety concerns with more unbiased data in order to implement this device.
Yeah, I mean, it could be useful as long as it can be reachable for the average communities in the countryside, if it is easy to use and if it doesn't impose any other threats or harms to the environment. I think it's a great idea. So for most of the countries today that have extraction of gold, the governments and the big extraction companies are shifting away from mercury entirely, and they are moving into cyanide as their main composite to extract gold. But the problem is with the middle size and the small size informal and illegal miners, where most of the mercury is going to be a problem. So in that case, it would be a big challenge to create this technology and to make it usable and easy to apply, because the challenge is to be able to reach out to all these distant places where all this gold mining is happening.
Our device can potentially be implemented to mitigate present contamination. But at the end of the day, the more important issue with methylmercury contamination is to eliminate the source of contamination. This relies not only on the collective effort by synthetic biologists, but also on collaboration with law enforcement and pollution policymaking.
You can try and clean that up on the back end, right, with new technology. And again, when we're in the middle of the problem, it makes sense to me to do that.